Okay, so hi, I'm Michael, and today I'm going to talk about using convolutional neural networks to analyze bacteriophages' DNA. But before we start, how many of you have heard about the bacteriophages? Okay, nice. So the bacteriophages are viruses that target bacteria. So there are these tiny microbes in your system that target bacteria. And don't be afraid of the virus part, they're actually pretty harmless to humans. And what makes them interesting is that they can target only specific strains of the bacteria in contrary to the antibiotics. So as far as the industry use goes, they're used in diagnostics to, for example, detect if a certain bacteria strain is present in a sample. They're also researched in medicine. So the antibiotics work kind of like bombs. They can kill both good and bad bacteria in your system, which isn't ideal. On the other hand, the so-called phage cocktails can focus only on the specific strains of bacteria that cause your actual disease, which makes bacteriophages a much more safer alternative. And this is also why they are used in agriculture to protect the animals for certain bacteria strains. So the overuse of the antibiotics is one of the potential causes of the emergence of the so-called superbugs, which are drug-resistant bacteria. And of course, we don't want drug-resistant bacteria causing new diseases out there. So again, bacteriophages are a much more desirable and safer alternative to the antibiotics. But of course, we cannot just use any bacteriophage strains right away. We have to make sure that they meet certain criteria, one of them being how they reproduce. So based on the reproduction cycle, we can detect two distinct types of bacteriophages, lytic and lysogenic. So the lytic bacteriophages kill the bacteria right away. At first, they attach themselves to the bacterium, then they inject their DNA, then they start synthesizing the proteins by kind of eating them from the inside out, then they assemble new bacteriophage organisms, and finally release, killing the bacterium altogether. So nature is pretty brutal. The second cycle is lysogenic. So the lysogenic cycle starts pretty similarly. At first, the bacteriophage attaches itself to the bacterium and injects its DNA, but then, instead of eating the bacteria from the inside out, it enters this kind of dormant stage where it waits for the bacterium to reproduce. And when the bacterium reproduces, it replicates not only its own DNA, but also the DNA of the bacteriophage, allowing it to reproduce with it. And finally, when certain conditions are met, the bacteriophages activates and enters the lytic cycle to kill the bacteria and look for the new hosts. Now, these two cycles might seem pretty similar because they both end up with the lytic cycle, but the consequences are vastly different. So the lytic bacteriophages kill their bacteria right away, and there's not a problem. But the lysogenic bacteriophages can move parts of DNA between the bacteria, which is a possible cause of mutations. Of course, not all mutations are dangerous, but then again, we don't want new mutated bacteria strains out there causing new diseases, so we only want to focus on the lytic bacteriophages when we make the so-called phage cocktails. Now, a little bit about the origin of the project. So recently, we went with a team of programmers and data scientists to a competition called Biohack held in Łódź. And the goal of the competition was to create an application within about 24 hours on one of the given topics. So the topic that we chose was proposed by Proteon Pharmaceuticals, which deals with bacteriophages on a daily basis. The goal of the task was to create an automated classifier that can tell whether a bacteriophage is lytic or lysogenic based on its genome, so based on its DNA. Currently, it takes up to weeks of laboratory work to detect whether a bacteriophage is lytic or lysogenic. So the hope of this company was this task that could be automated with a machine learning tool. So we actually managed to solve this problem and win the competition, and so I'll tell you how. But first, let's focus on data. So during the competition, we managed to crawl about 2,000 bacteriophage genomes, which means we had about 2,000 separate bacteriophage strains in the training set. The training set was pretty much balanced. I mean, there are a little bit more lysogenic bacteriophages, but not enough to cause any significant issues. What did cause some problems was the size of a single sample. So each genome could contain up to 200,000 nucleotides, which are 200,000 separate categorical features. Now, it might not seem like much for the biologists because even the bacteria strains have millions of nucleotides, but in terms of machine learning, it starts to be problematic. And a quick reminder about the representations of the DNA. So at first, you've got the sequences of nucleotides, and there are 4 possible nucleotides: adenine, thymine, guanine, and cytosine. Then triplets of nucleotides make up codons. Codons encode amino acids. And there's a bit of redundancy here because there are less amino acids than there are codons. So amino acids can be represented by multiple triplets of nucleotides. And finally, sequences of amino acids can encode proteins, which can be synthesized by the organisms. So in this case, when I'm talking about DNA as data, I mean sequences of nucleotides. So the genomes that we managed to find were these huge strings of A's, T's, C's, and G's. And when we look for publications that deal with DNA as data, they usually focus on a single species or a single part of a genome. So if you take any two humors, then the DNA will be mostly the same. There will be a few nucleotide differences here and there that you can extract as categorical features, but the rest will be mostly the same. In this case, we're dealing with multiple bacteriophage strains with vastly different genomes, so we couldn't just extract a few features and call it a day. We actually had to process the whole input sequence. We had to process the whole genome with our classifiers. And as I've mentioned, each genome could contain up to 200,000 nucleotides, and since there are 4 possible nucleotides, that's 800,000 features. And that's a lot for many classic machine learning algorithms, especially if they can't handle sequential data. So in order to fix this problem, we used convolutional neural networks. So convolutional neural networks were originally designed to process image data, but really they can handle any kind of sequential data there is as long as you prepare it properly. So what makes convolutional neural networks really good in this case is that they create this sort of reusable filters or kernels that look for patterns throughout the whole input sequence. So it's very important in the case of DNA because it doesn't really matter if a certain protein is encoded at the beginning, in the middle, or at the end of the sequence. We just care that this organism, that this bacteriophage can synthesize this particular protein. And convolution kernels could detect these features regardless of its position. But of course, we couldn't just use any image processing architecture out there without any modifications. So let me give you a high-level overview of our model. So at first, the model uses nucleotide sequences as input, so basically huge strings of A's, T's, C's, and G's. Then it performs a simple one-hot encoding vectorization. So for each nucleotide, we assign a unique vector that represents it numerically. So for example, A is represented by 1, 0, 0, 0, T is represented by 0, 1, 0, 0, and so on. This allows us to convert from this sort of string input type into a series of vectors or matrices that represent the whole genome. Then we pass the data into the first convolutional layer, which was specifically designed to handle codons. So as I've mentioned, codons are triplets of nucleotides, which means this layer looks at triplets of nucleotides to process them further. So the goal of this layer is to convert from this sort of sparse representation of nucleotides into dense representations of amino acids. Then the data is passed into the further convolutional pooling layers, which allows us to detect higher level features. So for example, the first layer could detect amino acids, next ones could detect proteins, and the final layers could detect series of proteins throughout the whole genome. Then when the convolutional pooling layers are done with the data, we convert it into the fixed size vector, which allows us to pass it into standard fully connected layers, which process data further, and finally into the output layer, which gives us probabilities for each class. Now, this might seem like your any image processing architecture except for the input type, but there were a few tricks that we had to apply in order to make this work. So at first, we had to use much wider convolutional kernels. So if you look at many popular image processing architecture, they usually only look at like 5 or 3 pixels at once, but in this case, it simply wasn't enough. Before we made sure that the convolution kernels are actually able to see the whole input sequence, so the last convolution layers are actually able to process the whole input sequence in a single step, we weren't able to build any sensible models. And I'm not talking about a few accuracy percents here and there. We actually weren't able to build any model that didn't always return 1 or 0, which of course isn't ideal. So you always have to make sure that your model does that. And we also use skip layer connections. This is inspired by the recent architecture. So skip layer connections is this very simple concept. Instead of placing your layers sequentially one after another, you allow for some arbitrary connections between the layers, which in turn allows the layers to handle both low and high level data. While it didn't exactly improve our scores on the validation sets by much, it did improve our scores on unseen data, which makes us believe this is very important to improve the generalization of the network. And finally, we did apply some expert knowledge. So sometimes it might seem like you can just kind of take the data and throw it at the model and somehow it works. Well, we kind of did that. But in this case, when we did apply some domain knowledge to improve the models, it did improve our scores. So in this case, we created the first convolution layer, which was specifically designed to handle amino acids, and it did improve the results of our model. So you always have to make sure that you understand the domain before tackling a problem. And of course, there were other issues, like the input was so huge that we had to use very small mini batches. But let me not bore you with that, and let's get to the results. So we managed to achieve about 97% accuracy on the validation set and 100% accuracy on the testing set. And before you say that this 100 accuracy sounds pretty suspicious, because I mean, kind of does, it's only because the testing set that we received during the competition was so small. It only had like about 10 samples or so. So we still expect over 90% accuracy on unseen data. And I mean, this is pretty much enough. Of course, we cannot replace the biologists just yet, but letting them focus on the bacteriophages that are interesting can significantly speed up their work. So the biologists only look for the lytic bacteriophages because they are the only ones that they can use commercially in the phage cocktails. So if we can confidently say that some bacteriophages are lysogenic, they can just kind of throw them out, filter them out, and use only the ones that we think are potentially lytic. But of course, the biologists won't trust us before they can understand what we're actually doing, so we had to focus on interpretability from day one. So at first, we used LIME, which stands for Locally Interpretable Model Agnostic Explanations. So LIME works by building a second, simpler model that tries to understand your original black box classifier. So for example, at first LIME generates these sort of modified versions of your original input that you're trying to understand. So for example, slightly distorted genomes. Then it passes the modified inputs into your original classifier to see how it reacts to the changes. And finally, it builds a second, simpler model that's much easier to understand and checks which features were important. So in this case, it didn't really work at all. I mean, LIME is pretty great for text, but in the case of DNA, it performed very poorly. So at first, it took ages to build, and I mean like minutes for a single input. But what's even more important is that the results were not reproducible at all. So if you build like 3 separate LIME models with 3 different seeds, you'd get completely 3 different sets of features that were important, which isn't ideal. So instead, we used class activation maps. So class activation maps is this neat algorithm that allows you to visualize activations of your actual model in relation to the output class. So what's really fun about it is that you can generate separate class activation maps for each output class of your model. So in this case, we're able to highlight both lytic and lysogenic features in the original genome. And of course, class activation maps are generated with your actual classifier, so they are fully reproducible and take much faster to generate. So the whole classification and the interpretation takes well under a second, which is enough. As far as future steps go, we really have to focus on the feature engineering. So currently we're using a very simple vectorization method of the nucleotides, but there are so many other ways that we can represent DNA. So for example, instead of sequences of nucleotides, we may try sequences of amino acids. We could also try building DNA embeddings. So embeddings is this cool concept for NLP, where you build vectorized representations of certain entities. So for example, if you build embeddings for words, and 2 words have different letters but have the same meaning, their vectors should be very close to each other. The distance between them should be very small, which represents their semantic meaning. But in case of DNA, we could build embeddings, for example, for proteins. And if the proteins have different structures but have the same function, their vectors should be still very close to each other. So this is something that I want to focus my current research on. And of course, we might try other architectures and other algorithms that can handle sequential data. So during the competition, we only had realistically time to test a few convolutional neural network architectures, but now that we can really focus on the problem, we want to try out other algorithms to see if either our solution is the best one or the most suitable one in this case. And finally, we're building a web application with Proteon Pharmaceuticals for all things bacteriophage. So it would have a huge bacteriophage database. You could upload your bacteriophages, get the classification, get the interpretation, and so on. I think it's pretty rare for a hackathon project to end up with an actual product, so this is something I'm really excited about, even though it's only part-time, but yeah, this is something I'm currently working on, so wish us luck. And thank you, that's all from me. If you got any questions, don't hesitate to ask.
First of all, thank you. I was just wondering, because my background is in biology. I was wondering, why couldn't you do something like use a BLAST algorithm to identify similar sequences and put a logistic regression on top of it?
That's what the biologists wanted us to do. But then again, we're a team of data scientists, so we kind of just used the tools that we knew. And it turned out that it's actually better than the previous approaches that they tried.
What was the previous approach?
Honestly, I don't know. I'm not a biologist, but they said that our model performs better than their solutions, I guess.
I just mean this because there are some very mature technologies like BLAST, which is basic local alignment. You just find sequences and find similar ones in known organisms, and you can.
Yeah, I get. But I think the argument was that sometimes even the lysogenic bacteriophages can, or the other way, the lytic bacteriophages can contain proteins that are used solely by the lysogenic bacteriophages, but they still don't perform the lysogenic cycle in practice, right?
Okay.
So it's not that easy. It can contain the proteins necessary to, for example, cut itself out from the DNA, but just doesn't use them, right? So.
Okay. Thank you.
Hello. That was a great speech. Thank you for that. Eye-opening, really. But I would have two questions, maybe. First, have you compared it against a simpler approach, like, for example, K-mer approach?
I'm sorry, what approach?
K-mer-based approach that you split the whole genome into K-mers, like 9 mers or 8 mers.
Honestly, I don't know any specific biologist tools.
That's not biological. That's rather like natural language. So you take just 5, 6, 7, 8.
Oh, you mean n-grams?
Yes.
Oh,
or K-mers. That's probably biological term. That's maybe why the confusion arose. So have you compared your solution against that simpler one?
I mean, we did use some simpler models like within the team, but this one performed the best. So I can't give you the exact metrics, but the convolutional neural networks compared to, for example, recurrent networks and other approaches were just the best. So
probably that's the matter of representation. And the other one, how can you assess the possibility of generalization of it? Because as you mentioned, and that's a very clever remark, that but for example, genomes of bacteria are 20 times bigger than that, and then it would require really huge machine learning models. And
that's why I'm counting on the DNA embeddings to actually help with that, because they can kind of reduce the dimensionality of the input.
Especially that it is said that 98% of genome is not used at all for coding. So maybe that may be I don't know, a point to focus on, to try to find what is really relevant within genome. But maybe that's for also for future work. Thank you.
Thank you.
So very nice talk. I have several questions. One is, have you accounted for because it's already known that some genes can overlap in bacteriophages.
Right.
Have you accounted for that or not? This is one question. I was also going to ask about K-mers, but it was already done. And another one is, what was the input? What is the desired input for the classifier at the end product? Because usually we sequence the genomes of bacteriophages or bacteria, but sequencing process is prone to errors. Yeah. So do you account for that or not? And the third question, have you accounted for different frames of codons? Because these triplets you read in the sequence
are different
they
for
are not
each.
at the fixed position, but you can read from the one side or another side, and they can be shifted. So have you accounted for that in the model?
So the convolutional kernels overlap. So in this case, it doesn't really matter where we start, because they would detect the features either way. And you have to remind me the previous questions, because I'm just...
So have you accounted for the overlapping genes. So
yeah, that's it.
And have you accounted for the sequencing errors? Because I suppose that the product should work on the sequenced genomes, and the sequencing of the genomes is prone to errors. So do you...
Currently no, but obviously since you're talking about it, this is probably one of the problems that we'll have to solve in the end product. So currently I'm just starting in this domain. I usually work in NLP commercially, so this is all new for me. I just wanted to share with you the project, and there might have been slight errors in the biology side of things that
That's correct.
Yeah.
That's correct. Thank you.
So any other questions?
Thank you.
Thank you. Thank you.